Hello and welcome to this study session with Caroline Cumberry. Today we're looking at the Biology AS Unit 1.1 and one of the first topics that you study at AS level, which is the structure and function of carbohydrates. So let's have a look at the essentials then of carbohydrates. What we need to know. So first of all, to appreciate that carbohydrates are organic compounds. Now, organic by definition means carbon-based or mainly carbon-based. I have a rule for deciding whether something is organic. Count up how many atoms that are there and in a particular molecule, and count up how many of those atoms are either carbon or hydrogen. And if the number of carbon or hydrogen atoms is over 50% of the total number of atoms present, then you can safely say that it is organic. So it must contain carbon and hydrogen, and the number of carbon and hydrogen atoms must comprise over 50% of the total number of atoms present. Usually it's pretty straightforward what's organic, because you're going to see a lot of carbon and hydrogen there. But okay, that is the official definition, or the way to actually discern whether something is organic or not. So carbohydrates are organic, just like proteins, just like fats. Carbohydrates contain carbon, hydrogen, and oxygen. Two main functions. First of all, they can be a source of energy, and that's probably what we're most familiar with from GCSE. They provide energy to both plants and animals, and a couple of examples of where we would see that. Sugars, starch, and glycogen are both ways in which we store energy. So number one, source of energy. The other thing they can do, which is less familiar from GCSE, is they can have a structural role. So cellulose is a carbohydrate. You would have heard of cellulose from GCSE. It's a component of cell walls, but it's less obvious that it is a carbohydrate. But we'll go into more detail on cellulose and other carbohydrates later on. Now, we usually categorize carbohydrates based upon their structure, and they categorize into three groups. So you can be monosaccharides, disaccharides, or polysaccharides. Now the suffix there, the end of each of those words is the same. The saccharides. Mono means one, sort of one unit, one saccharide unit. Di means two, two saccharide units, and poly means many. So you can see the polysaccharide is lots of those monosaccharide units joined together, and a disaccharide is two of those units joined together. So here's what I'll do throughout this this video, give you opportunities to fill in the blanks, just a little gauge of whether to just mean that you're doing a little bit more than just listening, because listening to me for a long time is going to be hard work. So you can pause the video and have a little go at filling in the blanks, and it kind of keeps you engaged a little bit more. But I'll go through the answers now. So carbohydrates, they're organic compounds containing carbon, hydrogen, and oxygen. Two possible functions: a source of energy or a structural role, number three. Fall into one of three types: monosaccharides, disaccharides, and polysaccharides. Okay, let's move on. So let's just focus now on the monosaccharides to begin with. So monosaccharides we refer to as the monomers that join together to form polymers. So a monosaccharide is the specific name of the monomers we find in carbohydrates. And a polysaccharide is the specific name of the polymer we find in carbohydrates. So the term monomer and polymer is not specific. It can be applied anywhere where lots of the same unit are joined over and over again. So lots of monomers joined over and over again to form polymers. But when we're specifically talking about carbohydrates, we don't need to say monomer, which is a general term. We say monosaccharide, which specifically relates to carbohydrates, and the same for polysaccharides. All monosaccharides have the same basic formula. So CH2O, all in brackets, N, where N is the number of carbons. So if you had 3 carbons, that would equate to C3, 3 carbons, H6, 3 times 2, and O3. If it was 6 carbons, it will be C6, H12, 2 times 6, O6. Okay, they all have the same general formula. It's not a question you're asked about a lot, though. You classify monosaccharides based upon how many carbons are present, that N value from the previous formula. So triose monosaccharides or triose sugars, any monosaccharide can be referred to as a sugar. In fact, any disaccharide can be referred to as a sugar as well. You wouldn't refer to polysaccharides as sugars. It's from GCSE really, or even lower perhaps, the idea that we have carbohydrates are generally divided up into sugars, which taste sweet, of which monosaccharides and disaccharides are part of because they taste sweet. And what we would say then at GCSE is sugars and complex carbohydrates. Those are the polysaccharides. They don't taste sweet, so we don't refer to them as sugars. So how we classify monosaccharides? So triose, think of tri meaning 3, like a triangle, 3 carbon atoms. The O's that you see there is synonymous with carbohydrates. We see that ending O's often when we're referring to, or always when we refer to carbohydrates. So triose sugars have 3 carbon atoms, and we find them in metabolism. It's something we cover at A2, and we just sort of got to park this for now and just say, what do you mean by metabolism? Metabolism is all the reactions that take place inside the cell or inside the body, and there's the only time at A level where we actually see a triose sugar is when we get to A2 in respiration and photosynthesis. Pentose sugar, like a pentagon, means 5, so 5 carbon atoms, and they're involved in the formation of nucleic acids. Now, nucleic acid is a whole unit at AS. Really, we're referring to DNA, which you're familiar with from GCSE, and RNA, which is, if you like, is like a sister molecule to DNA, very similar. And then hexose sugars, hex for hexagon, so 6. Example here being glucose, the one you're most familiar with, the glucose sugar. And those are the ones that actually the main source of energy, those hexose sugars. So of those 3, hexose, I would suggest, is the most important, but you will come across pentose again, and triose, you can park it for now. It's unlikely questions will be asked on that. Right, same thing. You are going to pause the video, but I will go through the answers as they appear now. So monosaccharides are the relatively small organic monomers that join together to form polymers. They have the general formula CH2O, all in brackets, N, where N is the number of carbons. And the classification goes into triose sugars with 3 carbon atoms, pentose sugars with 5 carbon atoms, hexose sugars with 6 carbon atoms, and these are the source of energy. Okay, right, so let's look then at hexose sugars a little bit more. So we know already that all hexose sugars share the same formula, C6H12O6, because we know by definition that we're applying the CH2ON formula. So they all have to have the same molecular structure, but they, molecular formula, but they differ in their structure. We call molecules like this, which have the same chemical formula but different structures, isomers. Now that's something you might have covered at GCSE in chemistry. If you've got the same number of each element present, same number of atoms of each element present, but they're arranged differently, we say they are isomers of each other. Glucose exists as 2 isomers. So there's 2 ways in which that C6H12O6 can be arranged and still be called glucose. We have an alpha form of glucose and a beta form of glucose. This is the alpha form of glucose, and on first inspection it seems like there's an awful lot going on. What we've got here is 6 carbons, and you see the carbons are numbered 1, 2, 3, 4, 5, and 6. If you counted up all the hydrogens, you'd count 12, and if you counted up all the oxygens, you'll count 6. That's alpha glucose. How do you tell it's alpha glucose? Well, the key here is to look at the OH groups that appear on this sort of bottom rung of carbons. You see those OH groups, look at the orientation of them. This first one on the left is pointing down. the next one is pointing up, the next one is pointing down, and the next one is pointing down. So the way I remember this, I read from left to right, and I see down, up, down, down. And that is just something I've remembered is my way of identifying whether it's alpha glucose or not. It is a little bit odd, this structure. You see this ring structure here of carbons, well, it's largely carbons, but there's an oxygen in the top right corner. This is not our sort of normal molecule that we think of, but this is your standard molecule when it comes to hexose, or structure when it comes to hexose sugars. We have to be familiar with using or being comfortable with representing these molecules in a more simple manner. So because organic molecules are always carbon-based and have lots of hydrogen, we have a way of representing molecules in which we say, if there's a corner, if you see an angle, we just assume there's a carbon there. So every time there's a corner on this ring structure, or indeed, on this ring structure here, we say there's a carbon there. We know that carbon can have 4 bonds coming from it. So if you find a corner with a carbon and you count the number of bonds coming from it, if there's less than 4 or fewer than 4 bonds there, then you know the other bond that's left. In this case, I'm seeing just 3 bonds on the right corner there. So there's 3 bonds. So I know, therefore, that the other bond must be going to hydrogen. Now, I should be able to add this, and I'll show this on here. Let's put this as a pen here. So you see this. So I know, first of all, that's a carbon. And I know also there's another bond there with a hydrogen. I could do the same down here. That's a carbon, because it's a corner, and there's only 3 bonds there. So that's a hydrogen. Okay, so that's what that simplified structure is meant to show. That's what it's trying to help you with, to draw things more simply, and it takes away sort of a bit of the noise, I suppose, more than anything else. By that I mean all of this. There seems like an awful lot going on this left-hand side, and it looks simpler on the right, but it requires you just to use these rules. Oh, there's a corner, so it's a carbon, and I can count the number of bonds to know where the hydrogens would be. But we're not really interested in the carbon and the hydrogens. We're more interested in the OH groups. And you can see this is much clearer on this side. How do I know it's alpha glucose? Because it's down, up, down, down with those 4 OH groups at the bottom. Now, there is another type of glucose, beta glucose, there I mentioned. So beta glucose works in, looks almost exactly the same. But if you just concentrate on these OH groups here, I'm seeing a down, an up, a down, and an up. So whereas with alpha glucose it was down, with beta on the first carbon, in beta glucose it's pointing up. Or whereas with alpha glucose, I say it's not symmetrical, or it's not even, because it went down, up, down, down. With beta glucose, I remember that it is even. Down, up, down, up. Now, I'm only referring to these 4 OH groups. There is another OH group on carbon 6, but we're just trying to find a way to remember which is which. This is representing beta glucose in the more simple way, and again, you can see the OH groups goes down, up, down, up. Okay, right. Same thing. You can pause it, but let's go through here. All hexose sugars share the formula C6H12O6, but they differ in their structure. We refer to these, number 2, as isomers, and exist as 2 isomers, alpha form and beta form. We call them alpha glucose and beta glucose. Right, so how do we go about forming a disaccharide? So to form a disaccharide, that's about taking 2 monosaccharides together, joining 2 monosaccharides together. So here is a molecule of, look at the arrangement, down, up, down, down. So this is a molecule of alpha glucose. I've named it glucose. If it's just named as glucose, we assume it's alpha glucose. To be beta glucose, you would have to name it, or it would have to be named beta glucose. Our sort of default form is alpha glucose. Here's another alpha glucose drawn next to it. So to form a disaccharide, we've got to join these two molecules together. So when this happens, it's referred to as a condensation reaction, and that involves what we call, any condensation reaction involves the elimination of water. Water is H2O. I've circled there 2 hydrogens and an oxygen. Those atoms are eliminated from this reaction, and what is left is those 2 molecules drawn the same way, but now I've got that oxygen that was not taken in the formation of water joining together those 2 monosaccharide units. That has a name, that bond. It's called a glycosidic bond, and they're specific to carbohydrates. Whenever you join monosaccharides together, it's called a glycosidic bond. In this case, when you join 2 alpha glucose molecules together, we form a disaccharide which has the name maltose. So the disaccharide is maltose. There's 3 possible disaccharides, or 3 disaccharides you need to be familiar with. You join glucose and glucose and you form maltose. If you form glucose and another monosaccharide called fructose, found in fruit, you form the molecule, the disaccharide sucrose. Sucrose is table sugar, the sort of sugar you put in your tea or on your cornflakes. That's your standard sugar you have at home. And the final one is if you take glucose and with another monosaccharide we're not familiar with yet, is galactose. Join those together and you form the disaccharide lactose. So you need to know the names of the disaccharides and which monosaccharides join together to form each disaccharide. You can go the opposite direction. You can go from a disaccharide to a monosaccharide, and that is referred to as hydrolysis. So instead of, or a hydrolysis reaction. So instead of a condensation reaction which joins together 2 monosaccharides, we have hydrolysis which separates 2 monosaccharides. Right, let's have a look then at polysaccharides. So there's 2 types of polysaccharides. So what we know is that they are polymers when you get lots of monosaccharide units joined together. And there's 2 main types: storage polysaccharides and structural polysaccharides. The storage ones, names you'll be familiar with from GCSE: starch, glycogen from GCSE. And structural polysaccharides, we talked about this earlier. So we have structural carbohydrates. They form 2 functions: energy, in which case starch and glycogen are a storage of energy, and structural polysaccharides where well, they're structural, and we have cellulose there. And another one that is not familiar to you, and that's chitin. So not something you study at GCSE. So let's have a look at the storage polysaccharides first. So starch is odd in that is rare we come across this, but starch is in biology. Starch is a mixture of 2 compounds. So it's the first compound that is a mixture of is amylose. So amylose, which makes up 20% of starch, it's just alpha glucose molecules joined over and over and over and over again in a linear fashion, in a line. It has no branches coming off it, and all you've got is glycosidic bonds joined over and over again. We say they're 1,4 bonds or 1,4 linkages because it's joining the carbon 1 to the carbon 4, just like I showed when forming the disaccharide. That was carbon 1 joined to a carbon 4, so it's a 1,4 bonds. There's another component of the mixture which makes up starch, and that's called amylopectin. Now, amylopectin forms the greater proportion of starch, 80%. It's just like amylose. It has loads of alpha glucose molecules joined together over and over with 1,4 linkages, 1,4 bonds, but every now and again we also get a 1,6 linkage, and that means you have branches coming off amylopectin. So if you look at this amylopectin molecule, we've got, if you like, this linear molecule here of alpha glucose molecules joined by 1,4 glycosidic bonds. We've also got a linear molecule here of alpha glucose molecules joined by 1,4 glycosidic bonds. But these two glucose molecules just there, they are joined by a 1,6 glycosidic bond. Anytime you see a branch, that is a 1,6 glycosidic bond. Anytime you see a branch, it's a single 1,6 glycosidic bond. You see, amylose doesn't have that. It's just a linear molecule that is coiled. We'll come back to that. There's no branches on there, so we say it's linear. Okay, as opposed to the branches we're seeing on a amylopectin. Glycogen, the other storage polysaccharide, is actually pretty simple to explain. It's just like amylopectin. It has branches, so it's not linear. It has branches, 1,4 glycosidic bonds with some 1,6 glycosidic bonds as well. Still alpha glucose. All storage polysaccharides contain alpha glucose. Okay, same thing. Welcome to pause, but I'll read on. Starch, a mixture of two components. Number one is amylose. Unbranched polymer containing 1,4 linkages only, and amylopectin, polymer with 1,4 linkages, but also some 1,6 linkages that give it a branch structure. Glycogen is sometimes called animal starch and is very similar to amylopectin. That's what we can equate it to. So you don't really need to learn the structure of glycogen. You just, by knowing amylopectin, okay, it's the same as that. What about with cellulose? So cellulose is a structural polysaccharide. And something we'll know from GCSE is that the job of cellulose is to give strength, support, rigidity to plant cell walls. We don't do any more than that. We just know that a cell wall is made up of cellulose. That's what we know from GCSE, but we don't want to look into any more detail. The difference between, or the first difference we know between storage polysaccharides and structural polysaccharides is that now with structural polysaccharides, they contain beta glucose. So we've still got a long chain of monosaccharides, but in this case, it's beta glucose. So you can see there, multiple beta glucose units, 1, 2, these are all beta glucose units joined together in one long chain. However, within each chain, every other beta glucose molecule is rotated by 180 degrees. So I've drawn a beta glucose molecule here. I've drawn another beta glucose molecule next to it, but you can see it's been flipped or it's upside down. Whether you say it's flipped 180 degrees or rotated 180 degrees, it doesn't really matter. Mark schemes will allow either. I think probably I prefer to use the term flipped. I'd say that's flipped over rather than rotated. But you can see, if we just focus on this CH2OH group, it's pointing up on this first molecule, and the one adjacent to it is now pointing down. And then on the other side of it is pointing down as well, and then it's pointing up. So you can see I've got points down, points up, points down, points up. It means then that when you join those molecules together, it's still with 1,4 glycosidic bonds. Okay, these bonds are still with 1,4 glycosidic bonds, but we've got a different orientation of both the CH2OH groups, but also all the OH groups as well. And these OH groups are significant to us. They make a difference, which we'll see shortly, these OH groups that we see all along there. Okay, and continuing on all of these, okay, and we see them as part of the CH2OH as well. Okay, we've got all these OH groups. But the key thing is, because you're, if you like, because they're being every other beta glucose molecule is flipped, the proportion of OH groups above or pointing up and pointing down is the same. It's the same. Okay, so what does that mean? Why is that relevant? There's my chain of beta glucose molecules. Those OH groups, equally pointing up and down, result in the formation of hydrogen bonds between adjacent parallel chains. So I've got a chain here of beta glucose molecules, a chain here of beta glucose molecules, and these are hydrogen bonds between the adjacent beta glucose molecules. I can add one there as well. Now, it's important we check our wording here. We must be referring to so what I've said in there is that the hydrogen bonds form between the OH groups or hydroxyl groups of adjacent parallel chains. What you cannot say is that there are hydrogen bonds between adjacent beta-glucose molecules. Now, if you look, there's a beta-glucose molecule here, there's a beta-glucose molecule here, and indeed there is a hydrogen bond between beta-glucose molecules. But the trouble is there's also a beta-glucose molecule here, and this is not a hydrogen bond between them. That's a glycosidic bond. So you've just got to be careful with your wording here, and the wording I've got on this slide is carefully chosen to be tight, that it's going to get you out of any trouble. So when you're talking about hydrogen bonds, you need to be referring to hydrogen bonds between chains. And I say adjacent parallel chains, even better, because the fact that you're saying they're parallel also increases your chance of picking up marks. If you're, I mean, it would be a poor question if you were asked what type of bond is present between two beta-glucose molecules in cellulose. I mean, that would be a poor question, because you could give two answers. You could say a glycosidic bond or a hydrogen bond. If they said what bond is present between beta-glucose molecules within an individual chain of cellulose, yes, you could say it would be a glycosidic bond. Or what's between two beta-glucose molecules in different chains or parallel chains, that's a hydrogen bond. But the wording there is key. And those hydrogen bonds we refer to as crosslinks. Okay, so the crosslinks and the hydrogen bonds are the same thing. Crosslinks are more of a general term. Anything which links across, we can refer to it as a crosslink. In theory, in different molecules, in biology and chemistry, whatever, we could refer to crosslinks in many different ways, but then we dial down into the detail and we see what is the crosslink there. Oh, it's a hydrogen bond, or whatever else, that whatever form that bond takes. In this case, the crosslinks take the form of a hydrogen bond. So there we are. Those are the crosslinks that give cellulose, the crosslinks that we see there. Those are what give cellulose its structural stability. Now, when you get lots of chains cross-linked to each other, it's referred to as a microfibril. And a microfibril is a term that appears on so many mark schemes. My advice would always be, even when you think microfibril doesn't answer the question, include it in your answer, because it's very common to see microfibril on a mark scheme. So my advice is always just to add a couple of as few words as possible at the end of your answer to just say, oh, collectively this forms a microfibril or something like that, because you often pick up marks for it, even when I think it doesn't answer the question. There we are. There's a microfibril. It's trying to give an idea of what we're talking about. So we got beta-glucose molecules joined together in a chain, lots of chains joined together because of the hydrogen bonds between them, and that forms a microfibril, or one part of a microfibril. Think of each of these little circles there being representing a few of these chains joined together, and then lots of them together, we get a microfibril. So there's the essentials. Okay, what's on the right there? Cellulose gives strength and rigidity to plant cell walls. Long chains of beta-glucose molecules. Within each chain, every other beta-glucose molecule is rotated by 180 degrees. Forms hydrogen bonds between hydroxyl or OH groups of adjacent parallel chains. It's these cross-links that give cellulose its structural stability. And when you do this over and over again, multiple chains, the structure is referred to as a microfibril. Right, so as before, what answers can you come up with for these blanks? Right, so cellulose gives strength and rigidity to plant cell walls. It's often marks for saying that cellulose gives strength to plant cell walls, even though I would class that as a GCSE idea, I think. Consists of long chains of beta-glucose molecules as opposed to alpha-glucose. And look at the word I've left out here. I've left out the first word, within, because I'm saying that word there, it's not a scientific word, but it just concentrates you on what you need to be referring to first, right? Just talk about what's going on within the chain. Every other glucose molecule is rotated, you could say flipped by 180 degrees. And this results in hydrogen bonds forming between the hydroxyl groups of adjacent parallel chains. Again, those words I'm missing out there are meant to guide you. They're waypoints, the things you need to include. Adjacent is not a biological word, it just means alongside each other. But I picked out those as being the key words when you're trying to explain or describe the structure of cellulose. And it's these cross links that give cellulose its structural stability. And then when multiple chains are cross-linked to each other like this, the structure is referred to as a microfibril. Okay. Lastly, let's then have a look at chitin. So chitin is new. It is found in insects, in the exoskeleton of insects. Insects don't have an endoskeleton, a skeleton within them like humans do. Their skeleton is on the outside, sort of think of it like an outer shell, if you like. It's very similar to cellulose. So once you know the structure of cellulose, chitin is pretty straightforward. You just got to make a few amendments. So there's cellulose. With chitin, some of the OH groups are replaced with amino acids. So I'll just go back one. You can see this OH group here, and this OH group here, and this OH group here. Am I right? Yeah, they're replaced with amino acids. So each of these green shaded areas, those are amino acids. Now, what does that mean? Not hugely different to what happened with cellulose. Those, let me go back to that a moment. Those amino acids can form cross links with each other. In the same way that OH groups formed cross links hydrogen bonds with each other. Okay, so we still get that same idea of strength as a result of the cross links between adjacent parallel chains. This here is referred to as glucosamine because you've got like a glucose molecule plus an amine or amino acid. Each of those is referred to as a glucosamine. You put lots of them together and we end up with like the equivalent of a microfibril, a mucopolysaccharide. So in chitin, we refer to the whole structure as a mucopolysaccharide. In cellulose, we refer to the whole structure as a microfibril. Okay, last one. So pause if you like. Let's go on. Chitin forms the exoskeleton of insects and has a very similar structure to cellulose. In chitin, some of the OH groups are replaced by amino acids, forming a molecule called glucosamine. I should say the first part of that, the idea that OH groups are replaced with amino acids, very common to see when you're asked about chitin. Questions on cellulose appear a lot, questions on chitin far less often, and when they do, it'll be the amino acid part that's most likely to be asked. And the whole structure referred to as a mucopolysaccharide. Now, a useful activity here when you're trying to look at the features of polysaccharides is to, I'm a big fan of trying to take the information that you've been given and try and order it, trying to get some order out of it. So comparing different features. And so this is an exercise I will often do with my students. I'll ask them to look at the information they've been given and say, well, how can I distinguish between them? So let's go through what you should have on here. So starch. First of all, what bonds we got present? So in this is a tricky one because actually we can't just say what bond, give a simple answer here because starch is a mixture. So we have to say, well, amylose is got alpha 1,4 glycosidic bonds. Amylopectin, the other component of starch, has got 1,4 and 1,6 glycosidic bonds. Now with glycogen, because it's like amylopectin, it's just got alpha 1,4 and alpha 1,6 glycosidic bonds, just like amylopectin is. For cellulose, well, that's tricky. What bonds are we talking about? Well, within the chains, it's beta 1,4 glycosidic bonds, but between the chains, it's hydrogen bonds, making those cross links. So I'm giving detail here because it's quite tricky just for me to be talking about what bonds are present when there's detail needed. And chitin is going to be the same. It's still beta 1,4 glycosidic bonds within the chains, and it's still hydrogen bonds between the chains. It's just between amino acids sometimes, rather than just between OH groups. In terms of shape, starch is a coiled structure, linear, you could say. Whereas, no, let me reword that. So amylose is a coiled structure and linear. Amylopectin is a coiled structure, but not linear. It's branched. Okay, it's branched. Glycogen is coiled and branched. All storage polysaccharides have a coiled structure. Not all are branched, because amylose is not branched, but it is coiled. And the idea that coiling makes it more compact, good for energy storage. For cellulose, bit of detail. So I got long chains of beta glucose molecules, but they're cross-linked to each other by hydrogen bonds between OH groups. Whereas for chitin, I've said long chains cross-linked to each other by hydrogen bonds between amino acids. You could say between amino acids and OH groups, but the key thing is there, the distinguishing part is between amino acids. What about function? Well, starch is all about energy storage. Glycogen is all about energy storage, very straightforward. Cellulose, its function is to strengthen cell walls. Chitin is found in the exoskeleton of insects. So see, we're talking about the same feature, but see how it differs with each of the polysaccharides. And then unique features, this is like, okay, things that we should be referring to here that don't really have a comparison in the other polysaccharides. So, okay, what unique features would we put in? Well, the fact that starch is a mixture is unique. Okay, you don't find that in any of the others. It's a mixture of two molecules, amylose and amylopectin, and it's found in plants then. Glycogen, not a mixture, so I don't refer to that, but it's found in animals. Cellulose, the idea that I could say found in plants, but I think key here is that the entire structure is called a microfibril. And chitin is just referencing to the idea that the OH groups are replaced with amino acids. The OH groups that were found in cellulose are replaced with amino acids. That's what makes it chitin, and the entire structure is referred to as a mucopolysaccharide. So nice little summary of the key features of the different polysaccharides there. And that's it. So I hope you found that useful. I mean, in all cases, whenever you did a fill in the blanks activity, what's a useful thing to do? I'll just go back to that here, and just something you can move on to, is you take those keywords there. So in this case, it was exoskeleton, amino acid, and mucopolysaccharide. And it's a really good activity just to take those keywords. You've done something very, I suppose, low level, very straightforward here. It's a fill in the blanks exercise. It's quite an easy entry level into this. If you were studying this for the first time, that would be your way in. But no doubt you've studied this already. So the next stage for you is to take those three words and see if you can put those words into sentences to outline the structure of chitin. So build upon those three words, use them as waypoints, and make sure you can then put them into meaningful sentences which describes the structure of chitin. But otherwise, that is it. I hope you found it useful. You'll find other videos on the Cara Cymru website, so please go and have a look at that. I hope you found that useful today.